This is a companion website to the VectorSurv Gateway. Some of these maps are publicly available. You can see up here these first three: Arbovirus, Invasive, Dengue. If you don't have Dengue, Zika, if you don't have a gateway login, or you want to share this website with anyone that doesn't have a gateway login, these three maps are publicly available. You can see there's little lock signs on abundance, resistance, and West Nile virus risk. You have to be logged into the gateway to be able to see those maps. So any data that's entered into the VectorSurv Gateway will get automatically populated to the maps and the graphs as a part of this website. Sometimes it takes a little bit of time to actually see that populate, but it should be fairly quick. Okay, so when you go to maps.vectorsurv.org, the first map you're going to see is this Arbovirus map. And you'll notice in here we have a lot of pretty colors, which are, they are Arbovirus positives that have been found in the state. This is in California because I'm in California. If you want to click other states that use VectorSurv, you can do so here. It'll take us over to New Jersey. You can also just move the map, zoom in and out, and you can see a picture of the US and where these different states are that are using VectorSurv and doing Arbovirus surveillance. So let's flip back to California. Okay, so to orient you here, we have all of these colors, which is our legend. So all of these positives that are showing up on our map correspond to our legend. If we're interested in only West Nile virus mosquitoes, we can turn off all of the other surveillance. A lot of these we don't have in California, so it's not actually doing anything on the map. So that's all my West Nile positives for this time period. So this is from January 2022 until present. Well, I guess future. I do want to point out that these dots on the map aren't exact locations. They have been modified so that you can't resolve exactly where this location is, but the spatial, you know, where the points are in relation to each other is preserved. So if we're interested in West Nile mosquitoes, just particularly from maybe July to October 2018, we can do that and get a small snapshot of what's happening. We can also hover over these dots directly to see where these positives are and how many there are. The size obviously corresponds with how many mosquitoes were positive in that time period. Also on our map, we can show surveillance. So this is showing that the surveillance effort. So of course, we're not going to find positives in areas where we're not doing surveillance, but you can see we didn't find, you know, maybe too many positives in this region where we were doing surveillance. And you can also show agency boundaries if that's helpful. And then finally, what I wanted to show you on this map is that we, I guess, two more things. When you zoom in, you can actually click on each of these dots and see at what point in time those mosquitoes were collected and what time they were positive. You can also draw a polygon around a region. So say I'm interested generally in this Roseville-ish region, not the greatest polygon, but we can see in a custom area what those positives looked like over time. And again, I can change this time period. It'll change my map and change my plot here. So I can see those positives over time and how they compare year to year. So I just want to point out again, when you do that polygon, the way to close the polygon is just to double click. Okay, finally, we do have a share button up here. So say I've oriented this map, I'm interested specifically in here, and I want to share this map. I can download my chart or I can download my map. We can also, there's ways to copy code or get an iframe if you want to embed these in anything. You can also just copy a link to this page just configured as it is if you want to share that specifically with somebody. So if I downloaded the map, it's just going to give me a PNG file directly of that portion of the map that's showing here. So that can be good to add to reports if you want. So that is the Arbovirus map. The next map we are going to review is the invasive map. And so this came about as we started seeing invasive Aedes in California. This map really started as a way for us to visualize that invasion. And we also wanted to be able to add remotely sensed data. We had a NASA project at the time. We wanted to add the remotely sensed temperature data to be able to model reproductive activity of mosquitoes and maybe be able to model where we would expect these to grow or what we would expect those populations to look like over time. So here's our map. In this one, we're showing Aedes aegypti and surveillance is in our green. So green is everywhere we've done surveillance, red is where we have found Aedes aegypti. You can also look at Aedes albopictus if you click on the blue up here. And you can also look at Aedes nodoscriptus. This started in California, of course. We know in some other states there may be some species like Aedes japonicus you're interested in, and those are conversations that are ongoing. And if you have other ideas, please send us an email at any point. I can't, say we will include everything, but it will definitely be a discussion. And hopefully we can include what is needed and will be helpful to other folks. So here we can move again. So let's look at Aedes aegypti. We can look at different times. So this is again showing early 2022 to middle of this coming year. We can look over time. So if we go back to just 2013, you can see we just started to have Aedes aegypti start being found in the Central Valley here in California. And if you go over time, it starts to grow. You can also click specifically on a location and a figure will pop up that shows collections, so general efforts, Aedes aegypti, where we've collected them. And then this blue line is our projected growth. So that's coming from that project I was telling you about earlier. So when we don't have surveillance or we haven't found aegypti, can we predict into the future what they may look like at certain times of the year, given different parameters? Okay, so let's make sure I'm telling you everything I wanted to tell you. On this map, this is something I didn't say. We do link to the mosquito control districts for each agency because this is your data and we want people to be directed directly to you if they have questions for you specifically. Okay, let's see. So we can click around and see what happens in different agencies, and we can look at that again over time, short periods of time. This will change, and it'll tell you when they were first found, last found, whatnot. Any questions on this invasive map? Could you explain what the projected growth percentage is? So this is based on a NASA project that was funded in our lab to take their remotely sensed temperature data, put that in with what we know about Aedes populations, and create a model basically for when we would expect to see growth periods of of that species of Aedes in this area based on the temperature. Great. So the next map we're going to look at is a dengue and Zika risk map. Again, these three are publicly available maps. You can see this is the week beginning last week. Very low risk, extremely low risk. It's not yet warm enough for a lot of activity to be happening. So let's move to an area of time that might be a little more interesting. So let's say late summer, early fall last year, we can start to see that this risk grows a bit in some regions. So if we want to know specifically what the risk is in, I believe these are by cities and census designated places. Someone could correct me if that's wrong. But if we're interested in the risk in Rancho Cucamonga, we can click on that and we can see what that risk looks like over time. That risk is based on all of these parameters down here that have been modeled to give us that output. So this could be really helpful if we started to see local transmission of dengue or Zika in our region. We can know, when do we expect risk to be high, and so that we can act accordingly. So this is based on vectorial capacity down here, if you're familiar. So this takes into account the density of mosquitoes. And this is not a perfect number, of course, because we don't know how many mosquitoes per person there are. That would be impossible to know. But we've basically set this at 5 being the max, and then it scales to that based on that growth rate that we just talked about in the last map. We pull the temperature ranges. Based on that, we know days between bites for these mosquitoes, the daily survival of those mosquitoes at that temperature range, what the incubation period of those viruses in the mosquito is, given those temperatures and temperature range, and then we get an output of a risk estimate. So if we move, later in the year, it totally drops off. If we go earlier in the year, we can see during August, September, which are peak Egypti times, is when we would see our highest risk. You can go look at another place. This place has high risk in the summer. And so you can use that to understand what risk in your region may be based on temperature patterns and Egypti growth rates. So the final 3 maps are only available to users with a gateway login. So if you are not logged in and you click on that, it'll say this map is restricted to gateway users only. Log into your account and then come back here and refresh the page. So I can click that. I'm not logged in, so I can log into the gateway. And then I can come back here and refresh the page. Voila. So we are going to look at our abundance map. Okay. So this abundance map shows you abundance of mosquitoes across your state, in your agency, and a breakdown of those. So we can look, at certain years. We can look at this is the whole species breakdown of what's been collected in California so far in 2023. A lot of Culex tarsalis. We can break this down by trap type. So if we're interested in BG sentinels, what are we finding versus what are we finding in CO2 traps? Go back to all traps. I can click on a region. So this is the Saciolo Mosquito Control District's agency boundaries. You can click there and see what's being found so far in 2023. It also gives us a bar graph up here of trap nights and what we find during those week-long periods. So starting to pick up as we're heading into warmer weather. This last week has been very warm and projected to be pretty warm this weekend. So let's see, we can also change the intervals if we want to look at month-long periods, or maybe, I don't know exactly what that was doing. Yep, month-long periods we can look at. We can change the end dates. Basically, I think it's great for everyone to play around with this. You can look obviously what's found in all agencies. You can look across states for what has been recorded so far. I think it's a really cool interface to get an idea of what's happening in each state. I think that's everything I want to show you. If we change the species up here, so let's say we were catching a lot of, I think it was limbs, or I guess we can look down here. A lot of Tarsalis. We can see per trap night, how many are we collecting? Oh, this is only in Sac-Yolo. If I go here, I can change this to full California. So we can see species per trap night. So that is our abundance map. So our next map is the resistance map, and this directly correlates with the bottle bioassays we went over earlier. When I click the resistance map, there's a lot of data that goes into this one, so give it a second to load. In California, we this is hooked up to the I can't even remember the agency now, but the pesticide use regulation people. And so that's why in California we can see this purple is showing us the pesticide use in California. I believe this isn't available to any other states because this is hooked up to California system, but other states do have the ability to see their bottle bioassays on this map. So for the purposes of this training, I'm actually going to turn off this pesticide use visualization. If you are in California, you can play around with it. You can turn on and off the different kinds of pesticides that have been used. I do want to point out that this data is quite old. This gets updated when the pesticide regulation folks or pesticide use folks update their data for us. That can take some time. So this tends to be a couple years at least behind. And as you can see now, almost 5 years behind. So I'm going to turn that off for now, and we're just going to focus on the bottle bioassays as this pertains to folks in other states as well. So for our bottle bioassays, we can see bottle bioassays over time. We can turn on and off which species we're actually interested in. So say we are only interested in different Culex species. We'll just leave all the Culex and turn off everything else. This one includes colonies. We'll turn that off for now, sure. You can also play around with which pesticides or insecticides you're interested in. As you can see, as we turn these off, some of our bubbles disappear because those were tested for deltamethrin resistance, for example. You can also change the scale of the coloration here. So this is set to, I think, the CDC standard to view the mean knockdown at 30 minutes. You can change that so our scale will change. If we want to look at mean knockdown at 90 minutes, our scale will change versus if we want to look at 15 minutes here. So that's up to you what you're interested in. The default is set at 30 minutes. So when we go in, we can click specifically on any of these points and we can look at what's going on there. So we click add or subtract all to the chart, and then in the chart we can see what's happening here. So we didn't get very good knockdown with our 2018 Yorkshire and Bowling Green Culex pipiens in this region. If we want to compare that to what's happening elsewhere, we can add this. See, we got really good knockdown of this Seymour Park pipiens. So we can add or subtract anything we want to our maps, or to our figure here, to be able to visualize what's happening. And so these are showing us species, amounts, insecticides, years, locations. And if we toggle over this, the legend here, we can see, which one we're referring to specifically. And that can be helpful if we add a bunch of stuff to our map. It can be really helpful to be able to toggle over because it can get a little bit busy. But this will give you a snapshot of what's happening and in any area where there is bottle bioassay data. And this has really come a long way in the last years. People are doing this a lot more, and it's great. So we're looking forward to having a more robust view of what's happening in the state. And of course, it helps you to understand if the products you're actually using are working in your regions, which of course are important for control. Our last map, which is a bit of a big one, is our West Nile Virus Risk map. So this map, I think it says here. So this map is based on the risk assessment model found in the California Mosquito-Borne Surveillance and Response Plan. So this gives us this scale here based on abundance, infection rates, vector index, converts to this scale so that we can see what's going on in our state. Later today, when we go over tools, we will talk about our risk model tool. And that is the same model that's feeding into this map. So we'll get a little more detail on how this model is actually working later this morning when we review that. But I wanted to review this as we're going over the maps now. So we can look, obviously now there's pretty normal low risk as season is just starting over here. If we go back to a time period like we're in 2023, so it won't let me do that. Up here, you can toggle to different years. If we look, and toward the end of the year, we can start to see more risk. I'm not going to go over the parameters of how this risk is fully determined on this map yet, because we will go over that. But I will show that you can look, in different species, what our risk is for West Nile. We can look specifically in an agency. So if we're interested in Sac-Yolo and understanding what that risk is, again, give it a second because there's a lot of data that goes into this map. We get outputs here. So this is saying what our overall risk is and where those are coming from. So this takes into account if there are sentinel chickens that have seroconverted in the region, if any dead birds have been positive, mosquitoes are infected, how many mosquitoes there were, and then environment, I believe it's temperature here. And then we also get this other sort of spaghetti plot output showing us what the risk is over time in Sac-Yolo in 2022. We can again change to different years. And then we get another output of the risk. Do you want to point out that 0 here is no data, not no risk. So no data has shown that there are any chickens that have seroconverted, but that doesn't mean that that hasn't happened. We just haven't captured that. We can look specifically at each of these types of risk if we are interested, but we do get a nice graphical output of all of them here. Again, with all of our other maps, you can share, you can download the charts, you can download the map, you can get a PDF. You can embed it in your website and get an iframe also to embed. Great, so that is the overview of our VectorSurv maps. I love our VectorSurv maps. I think they're great. I think they're wonderful visualizations, good snapshots of what's happening at any point in time. And our maps get better the more data that everyone adds. So thank you for doing that. And yeah, let's keep going. Anything folks online want to add or questions about our maps?
One thing that I'll just note quickly is that if you check some of the maps and see that your data isn't represented there, an example of this is like the abundance map, if you want to click over there.
Yep.
Is that some of the maps are dependent on us knowing what your agency boundaries are. And so this map is an example of that. So this is the boundaries for the agencies that submit data through VectorSurv. So if you're not seeing your data there, let us know and I'll let you know what type of information we need from you to get your agency boundaries within the VectorSurv database.
Great. Thanks, Lincoln. Yeah, and you can email help@vectorsurv.org to do that.